And this lecture is Chapter 3, Basic Principles and Concepts, Immunology and Pathology, Innate Defense Mechanisms, Part 1. When it comes to innate defenses are going to be our defensive systems, us eukaryotes, us multicellular eukaryotes, not just us mammalians. This is going to be a series of systems, cells, tissues, and whatnot that are there to defend the organism from invasion by other organisms. One of the biggest defenses we have, your skin, your integument, okay? Keeps stuff in, keeps stuff out, as long as it's in one piece, as long as there are no openings, no breaks. So the epithelial lining of our epidermis, the dry, dead, keratinized cells that we're constantly shedding, keeps stuff out. Now, is it impenetrable? No. There are different fungal parasites. There are different worm parasites that during their larval stages have the ability to burrow through skin. When we look at the integument, we also got to keep in mind the GI tract. Okay, our gastrointestinal tract is a series of layers of endothelial cells that are tightly bound together to keep anything from leaking in, anything from penetrating through. Again, is it foolproof? No. There are going to be different parasites in their larval stages have the ability to burrow through. There are different parasites like whipworms that have the ability in their adult stages to burrow into the underlying tissues to anchor themselves in place. The innate defense system has abilities to talk to themselves, to crosstalk, to send signals back and forth. As we see here, as you look here and you see here, this figure gives you an idea of just some small subset of some of the different receptors that are going to be on the surface of many of these types of cells. When you look at them, you'll see that the receptors, the artist has drawn them looking in different shapes. Not only are they in different shapes, the different shapes are to denote different protein motifs, different ways the proteins that make up that receptor, how they have folded, because remember, shape dictates function, so they're different shapes, different functions, and you got to keep in mind that those different receptors that we see right here are all antennae. Antennae that stick up and are looking to bind to one and only one signal. Some form of cytokine or chemokine here, or interleukin, or TNF alpha, TNF beta. Some sort of signal that the cell will then be able to respond to. Because these cytokines and chemokines, these are going to be protein or hormone-based signals that allow for one cell to talk to another cell. First cell is going to just release these signals out into their environment. That environment being, just under your skin, in the dermal tissues. Maybe it's in a muscle. Maybe it's in your liver, wherever. The first cell is going to release, and that signal is going to slowly diffuse through the tissues. As it diffuses through the tissues, it's going to form a gradient. Other cells when they come into contact with that signal, that cytokine, that chemokine, that protein, the polypeptide, the peptide fragment, the hormone, whatever, are going to respond. And those cells are then going to migrate up the concentration gradient, because if they get up that concentration gradient, they're going to end up at the first cell that released the signal because something told that first cell to do that. A bacteria, a virus, or some form of a parasite. Now, there's just a small list of cytokines, chemokines, things like that. It's not a great extensive list. There's only a few dozen. I'm not going to make you memorize them because this isn't an immunology class, so why would I do that? But what I am going to sit there and tell you about these is that the short list has a very broad level of what it can make other cells do, be dependent upon which receptor, which cell that receptor's on, where is that cell located, and all sorts of different things like that. So when you look at this table, you'll see that there are different cytokines, different chemokines that will be released by different cells, and they will have an activity on different cells. They will cause the cells to do different things. Because when you look at this picture here, what you see is that the receptors, remember from intro cell and molecular cell biology, whichever ones you've taken, that these receptors are transmembrane proteins or glycoproteins. The signal binds to it, causes a change in the cytoplasmic region of the receptor, which will then trigger some form of a downstream event, as we see right here. You see at the surface, the cytokine binds the receptor, causes the receptor to change shape, triggering the beginning of a signal cascade, a signal pathway in the cytoplasm of that target cell. Here, this pathway is what's referred to as the junk stat, JNK stat. It's a well-published, well-studied pathway that ultimately will activate transcription factors, and we see the transcription or the expression of a gene. Here's another example showing that the cytokines will bind a receptor, and it will trigger not just one pathway, junk stat, what we saw in the last one, but it can trigger the activation of multiple pathways in the same cell, leading to the expression of new genes, as well as the release or the activity of other cellular processes at the same time, all due to one signal. Now, sometimes it's not a cytokine or a chemokine that will activate one of these innate cells. Sometimes one of these cells will just come into contact with something that is not human, something that is not mammalian. We have specific receptors on the surface of our macrophages and neutrophils and a few other cells. These receptors are referred to, as you see right here, pattern recognition receptors. These receptors respond to specific patterns in protein shape or the sugars if it's a glycoprotein, or the sugars if it's a glycolipid. These sugar and protein shapes are known as PAMPs. PAMPs are going to be shapes, three-dimensional shapes made of sugars, chains of sugars, or amino acid chains that will not be found in mammals. They're going to be found bacteria, viruses, yeast, fungi, parasitic worms, but not mammals. So since they're not found in mammals, there's different receptors for each of those kinds. Because if you look right here, in this figure right here, each of the different colors is representing a different type of PRR, pattern recognition receptor. Each of those different receptors is going to bind a different type of PAMP. A special type of bacterial protein, glycoprotein, a special viral lipid, a fungal glycoprotein, or maybe a glycoprotein that's only going to be found on a parasitic worm. Once those PRRs are bound by their PAMPs, they're going to trigger, as you see right here, these different pathways. These different pathways are going to lead to different organelles within the cell, causing for, increased metabolic activities, increased reactive oxygen species, or different types of oxygen metabolites to be made because that macrophage has run into something not mammal, fungal, parasitic, whatever, and it's going to begin getting ready to phagocytize and kill. So it needs lots of oxygen species to inundate and damage. It's going to need more digestive enzymes in vacuoles. Going to also need to upregulate certain gene expressions to increase enzymes, the number of enzymes, to increase certain proteins for different metabolic activities. Another type of pattern recognition that we see, this one can be mammalian. Complement. Complement is a serum protein. Remember, serum proteins are proteins that are floating in your blood. It's a collection of proteins that are floating in our blood that, when activated, they literally fall like dominoes. Boom, 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 boom. The first domino cleaves the next one. That domino cleaves the third. That cleaves the fourth and fifth, and so on. Except instead of calling them dominoes, we call them C1, C2, C3, all the way up through C9. Anything that's non-self, these things will bind to. 1 cuts 2, 2 cuts 3, 3 starts a fast cascade of 4, 5, 6, 7, 8, 9. And by the time 9 gets activated, it's 9's punching holes in the plasma membrane of anything not us. Bacteria, viruses, parasites, fungi. The complement, as I said, this is going to be immune response, blood proteins. Blood proteins that are going to bind to the surface of something, not you. And as they're binding, they're clipping, acting as a protease to cut and clip the next step. C1 clips 2 and 3, 3 cuts 4, 4 cuts 5, 6, 7, and so on. The fragments that are also now binding to the surface of something, not you, those fragments can now interact with, as we see here, different receptors, scavenger receptors. Turns out things like macrophages, neutrophils, have a receptor that's going to bind anything not you. Anything not you or you but damaged. scavenge. Anything that can bind to it, the macrophage will then try to kill it, unless it's something living cell that's you, which tells it to go away. Don't worry how it does that. If you really want to know, take immunology, take cell biology. Complement receptors, those fragments that are binding as they're being clipped, they're binding to the surface of something not you. Well, turns out there's a receptor for those too. It's a specific pattern, those complement fragments, so there's going to be a pattern recognition receptor just for them. And then there's what's referred to as toll-like receptors. Toll-like receptors are a special class of receptors. These are some of the oldest receptors that we can think of, that we can find. Why do we think they're old? Because we find them in many common ancestors going back. When we look at phylogenetic trees and try to trace back with modern day organisms, trying to figure out what's the oldest organism, we find these in insects that we don't think have, evolved too much from, 100 million years ago. But we find them in the modern day versions, and we also find them in pretty much every other living animal. And then other defenses, IgA type of immunoglobulin, also known as an antibody. There's lysosome, which is an enzyme that goes and lyses bacteria. So you're going to find that being secreted into your, around your eye, into, the tears. You're going to find it in your mouth. You're going to find it, genital urinary tract, different places like that. Stomach, extreme low pH of the stomach means that most parasites, if they're ingested, are going to be digested along with whatever they're in, whatever fish or, mammalian musculature we're eating that those larvae or eggs are in. Once they're in us, they're also going to be digested away. Things that are referred to as interferons. This is a class of chemokine, cytokine right in there that's going to tell cells to kill what's ever inside of it, be it a virus, a bacteria, or some form of intracellular parasite. Think Plasmodium. And then a thing called tumor necrosis factor, TNF. I've mentioned this one already. This is a cytokine that's going to tell all cells it comes into contact with, get ready to kill. There's an infection about. Inflammation imminent.